Live streaming is on.
Thank you, Zeb. So, good afternoon, and there are some new faces as well. Now, this is the recreational part of MIWI. So, we are not only here to speak about science, but also to be recreational, let's say, I don't have another word, and to recognize as well the work of others. So we'll have a session here with some official speeches from us, from the organizers, from Bart and Amit from the MIWI specialist group as chairpersons and secretary. And then we will have the awards. So you are impatient to know who are the early career researcher awardee of this year and who is the Arden Locket awardee of this year. And maybe you're also impatient to know in which setting we will be in 2 years, in 2023. There will be also the announcement of the next conference. So let me first warmly acknowledge you for all this interaction that we have so far. We are not yet finished with the conference, that's clear, but since we need to convey the messages to most of the people worldwide, we choose to make the official speeches now and not this evening at the end of the conference. So it's a bit the same slides that I had at the beginning. So it was just a pleasure to be in this room here in Delft. So thank you so much, Nicole and Romy, Eric, and all the colleagues from the Senate Salle for helping us with the techniques, for accommodating us in this setting, and also Zeb from Dryfta for all the help that he provided. So a round of applause to all the administrators of this conference. Thank you so much. Then the aim was really to link microbial ecology and water engineering at the confluence of research, education, and practice. We see here again my former master student Nina Gupse, who was doing a master thesis between Delft and Aalborg. Actually, she went also by pair to do some sequencing analysis at that time, amplicon sequencing. And the aim is to bring our young professionals out of the desk from the classroom to the lab to think further on wastewater treatment plant and other water systems that we have covered during the conference. You see here David, who presented before, and Marta, who just wrapped her PhD thesis on purple phototrophic bacteria, and Jenny from the wastewater treatment plant Harnaspolder. And the aim of the conference was really to bridge how can we bridge the scientific findings that we have in the lab with enrichment cultures, with sequencing technologies, and other types of processes that we investigate with the full scale. How can we bridge that? And we thought that DNA and other informational molecules like RNA, proteins, metabolites, or lipids, maybe you are interested to look at lipids in an organism, how can it bridge and give information for the plant engineers or the operators of water systems in general. So we covered different topics from wastewater to drinking water via industrial water, groundwater, surface water, about microorganisms, method, pollution control, resource recovery. We want to dig into the processes. It can be the microbial processes or the engineered processes, process engineering, education. We will discuss much more on epidemiology and health this afternoon. So we started on antimicrobial resistance this morning and how we can also reuse water. So the aim was really to dig into these complex microbial communities. We have seen the complexity of microbiomes, the complexity of the methods, the number of methods that are available. It can be from computational to bioinformatics to analytical methods and biotechnological methods in the lab that we can apply. So everything relates to the research question, the investigation question that you have to design your plan. And maybe let's try to avoid too many long-term experiments in the future of 355 days, but maybe shorter experiments that can be replicated, reproduced with controlled experiments to really see a difference between what you measure currently in your system versus what are the baseline conditions. But we need for practice to have much more longer term studies, and having maybe direct investigation in full-scale wastewater treatment plants can be very interesting, such as what Nina, for instance, is doing with N2. So these were the learning objectives that I formulated at the beginning of the conference. Was asking you whether it is aligned with your expectations. And yeah, so that you can address yourself if the conferences delivered you the right messages that you wanted to have, or what were the surprises as well, and, on top of the expectation that you had. Definitely, we need to develop roadmaps to implement all our approaches together at the confluence of microorganisms and water systems. So there will be still 2 sessions this afternoon. Does that mean that after this talk we should go home and stop? 2 nice sessions on water quality, health engineering and education, and on wastewater or water-based epidemiology and risk assessment, the keynote lecture from Adei Desta from Ethiopia, and from Colin Notown from USA about the COVID-19. I would warmly like to acknowledge all the conference team. I already mentioned Nicole and Romy, Eric and his team before. I would like to thank Mark for all the support in the discussion that we had in this organization, and also the nice chairing of the session. I would like to thank Nina, G. I saw G before. I don't know where she went. She was here just passing by. Sergio, Francesc, and also Timmy, who help us to critically think about what is best for poster platforms, etc. It's clear we can do always better, but we did our best with what we had in hands, and I think we still nicely succeeded to convey a conference to the MIWI community. So let me here really warmly acknowledge Nicole and Romy. I don't know if Romy is here. You would have to... so I will wait. Okay, I have a present for you as well. Thank you very much to the MIWI Management Committee, to Bart and Amit for all the support as well, critical thinking on how to organize this conference online or in a semi-hybrid setting at different dates, 2022 and 2021, as you explained. And I'm happy that we were able to bring this conference in Delft and to broadcast it to the community. Thank you very much to all the keynote speakers, and I think it was very nice that we had this assembly of speakers here. Every session was led, and I think this is quite nice when you have an online setting. If delegates are not always able to join every session, at least there are some sessions that are self-contained. You have a keynote, you have 5 presentations, you have a roundtable discussion. And that, I think, it's nice in an online setting. So what we should not do when we do an online conference is to try to replicate exactly what we do in on-site conferences. And we need to have flexibility. It's clear there are some technical issues sometimes, but we have to act professionally. Thank you so much as well for the participants who encouraged us with your messages per email or in the chat box of the conference. It's very much appreciated. Yeah, with this, this is the end of my talk. I will wait that Romy comes back also to give a present to Nicole and Romy, and also to the PhD candidate who help us all through. Thank you so much. Now I will give the word to Bart and Amit for the award session, and some words from MIWI and some words for the MIWI awardees. Put that down. Yes, I had quasi forgot. This, you are just okay. You can.
Okay, good. Well, hello everybody. On day 3 of this MIWI 9 or MIWI 2021. I hope you enjoyed it as much as I have. And this is an exciting session because this is the time where we acknowledge people both early in their career and later in their career as being respectively promising and having established themselves as real pioneers in the field. And we also announced the next conference. But before I acknowledge the awardees, it's also good to reflect on the fact that we lose members of our community once in a while. Life is short, and it's good to also just take a minute to reflect those people who have been instrumental in the past in this field, way before it was called microbial ecology and water engineering, when it was still called activated sludge population dynamics, which was the early name of this research group. And I just want to spend two moments remembering two greats in our field, Professor David Jenkins, who passed away earlier this year. And many of you know David Jenkins. maybe from Snowick and Jenkins, a well-known chemistry book, or probably even more importantly from his work on many things that relate to microbial ecology. But one topic that he really worked on for many decades is the issue of flocculation. And so he also has this nickname of Flock Dog. And so he had a very long career, 6 decades. Most of us won't get there. And if you want to know more about David Jenkins, if you don't know yet, we actually had a remembrance written in one of our newsletters, which you can find or in the newsletter on IWA Connect. In addition to David Jenkins, sadly enough, as we were actually writing the remembrance of David Jenkins, David Stencil was contributing to that and passed away at that time. This was very sudden. While David Jenkins was a professor at UC Berkeley, as you may know, Dave Stencil was a professor at the University of Washington for many years. And he was also quite instrumental in our field and certainly worked intensely on the whole topic of nutrient removal throughout his academic career. Another remembrance for David Stencil was written, again, to be found in our newsletter or on IWA Connect. So this happens, and I think it's important that we don't forget the people who came before us, and that we don't forget to look at the literature that was published more than 10 years ago, once in a while. So on a more happy note, let's talk about the awards, the MUI Awards 2021. So every 2 years now, as our conference becomes biannual, we offer 2 awards. For that, we have an awards committee. It's not just me who makes these awards and these decisions. That would be much more efficient, but much less correct or appropriate. And so this is actually to give you an idea about the committee that looked at all of the nominations that were made, and we had quite a few. And our committee is, we believe, representative of the members of our community across the world. No, David did not get an award this year, but David was actually the first recipient of the Early Career Researcher Award, which he got in 2019 at MUI 8 in Hiroshima, Japan. This is an opportunity for me to actually acknowledge Hazen and Sawyer, a consulting company in the US who actually provides the financial support for this award. And just so as you can read here, the Early Career Researcher Award recognizes early career IWA members who are making promising contributions to microecology and water engineering, the community, through both research and service. David got the award in 2019 and organizes the current conference. So it is my sincere pleasure to announce the awardee this year of the IWA MUI Early Career Researcher Award, and that is Dr. Janwa Go, who is at the University of Queensland in Australia. Congratulations, Janwa. Much more applause. And hereby I give you virtually your award. And this then allows Janwa to make his presentation. If somebody can make that happen.
Yeah. Can you hear me, or can you see me as well?
Yes.
Wow.
Do you have your slides to present online, or you prefer that we launch your pre-recorded version?
I haven't our present online. I have a PPT. I will present later. So first, I would make a few appreciation. So I would thank International Award committee endorse my nomination. I also would like to thank all of my nominators, like Professor Black Linda Hall and Professor Lang Huaiman and Professor Zhi Guoyuan for their recommendation. So this is really like a big surprise for me, and also is indeed a big encouragement for myself and my research team during such like a pandemic challenging time. I feel very encouraged and supportive got such award. I would thank all the hard working from my team members, all the tremendous support from my family members. Like with their without their support, it's not possible to receive such award. Thank you very much. So one story I want to share with all the PhD students or even ECR, actually, I'm a big fan of the ASPD and MIVI conference. So when I was a PhD student, I submitted many abstracts to our ASPD, even the MIVI later, but unfortunately, I never get a chance to present my work through the oral presentation. But eventually, today I got the chance to talk through my research. So that's why I want to say, look, to be persistent as a researcher, like no matter you are a PhD or ECR or even like a master student, to be persistent. So sometimes your paper will be rejected or your grant will be rejected, but if you keep trying, so one day you will become a better you. So this I want to share with all of our young research and PhD students. So with this, I would share my PPT. Give 1 second. I have my PPT here. Let me find my slides. Can you see my PPT now?
Yes.
Okay, let me switch to the presentation mode. Okay, so I hope you can see. It's clear for everyone?
Everything
Hello?
seems perfect, yeah.
Okay, thank you, David. Yeah, today I will share one, my research direction is also related antimicrobial resistance or antibiotic resistance. So we have this session like this afternoon already, but I would talk through a little bit about our recent study, which is no antibiotic pharmaceuticals also contribute antibiotic resistance. So here I give like a subtitle is visualizing the invisible. What does that mean? I will go through and I hope you can get some like answer once I finish my talk. So we know AMR already. Like AMR is a big concept. It means the resistance of microorganism to an antimicrobial agent, including antibiotics, antivirus, antifungins, even other like antimalarials. So this is a very big concept. But the most important one or dominant one is antibiotic resistance. This is very, very serious. So here I have two jargons to want to define at the beginning. One is ARB. So for someone you are not familiar for AMR topic, so ARB means antibiotic resistant bacteria, and ARG stands for the antibiotic resistant genes. So how serious it is? Actually, I suppose you have gotten few numbers already. Here I would repeat again. So every year, around 700,000 people will die due to the multiple drug resistant bacteria, like infection caused multiple drug resistant bacteria or superbugs. So if no action is taken now by 2050, so how many people will die? Around 10 million people. So before the pandemic, so many were trying, look, this number could be overestimated, but now hope like a majority of people would believe this could be next epidemic if no action is taken now. So that's why this is a very, very serious issue for our public health. The second question I want to address, so how does AMR emerge and spread? So actually, they have 3 major pathways. So first one is mutation. So we know under exposure of antibiotics, some radical will be generated or some reactive oxygen species will be generated, such as OH radical or even the superoxygen radical could be generated. They have very strong oxidation ability. They could damage DNA. They also can trigger like a SOS response. So as a consequence, so bacteria will have some genetic mutation happened. So then some sensitive bacteria will become ARB due to the exposure of antibiotics. The second pathway is horizontal gene transfer, short as HGT, so including 3 suburb pathway. The first one is conjugation. So that means when we have a donor cell, they carry some like mobile genetic elements such as a plasmid. So then we mix donor and recipient. Recipient is a sensitive bacteria. So if we mix them together, so somehow or spontaneously, some like mobile ARG or plasmid, they could transfer from donor to recipient through some like PD-based bridge. So they can then the recipient cell will become ARB. So this is conjugation. So for the transformation means, so under certain condition, cell lysis is happened, like under like disinfection or even some other very stressful condition, DNA will be released. But some DNA or free DNA, they still carry ARG. We call the EARG, extracellular ARG, or free-living ARG. So some ARG that they could be swollen or uptake by recipient bacteria. So then that recipient could integrate ARG in their genome, then become the ARB. The third pathway is the transduction. So transduction is a little bit unique. So they rely on the bacteriophage. So bacteriophage, like shuttle bus, they can transport ARG from donor cell, then inject that ARG into the recipient cell, so the recipient will become the ARB. So this is a 3 suburb pathway related to HGT transmission. The third one is very straightforward to understand, is a VGT or vertical gene transfer. So this is very straightforward. For example, we have a parent cell. If the parent cell carry the ARG, they could pass ARG to the daughter cell or generation by generation. But somehow some ARG could be lost due to the fitting cost. Okay, so now, so as we know, the misuse or overuse antibiotics caused transmission or promote spread of antibiotic resistance. So the question we want to address, in addition to antibiotics, are there any other invisible trigger factors we have overlooked? So if so, what are they? So one idea pop out in our mind is, okay, so how about non-antibiotic pharmaceutical? So we know in medicine market, non-antibiotic pharmaceutical, they take account 95%. So while antibiotics just 5%. So usually if people uptake that pharmaceutical or even common use drugs, somehow this pharmaceutical will end up in our wastewater system as micropollutant or as PPCP usually we call. So look, the question here we want to ask, is any interaction between the non-antibiotic pharmaceutical and AMR? So this is our knowledge gap. So that's why we built up the first bacterial model is a conjugation model. So what we did, we choose RP4 plasmid, which is a conjugative plasmid. So that RP4 plasmid carries 3 antibiotic resistant genes, against tetracycline, calamicin, and ampicillin. So that is RP4 carried by E. coli. So this is E. coli donor. We also choose 2 recipient bacteria. One is the E. coli without any plasmid, but they are very tolerant to another antibiotic, which is CHL chlorphenicol. So we also choose Putita, Pseudomonas putita, as like another species recipient. So we mix them together. So you can imagine spontaneously that plasmid could transfer from donor to recipient. So then the recipient, oh, sorry. What happened?
Eric, there is a problem with the projection.
Okay, why am I lost my PPT? Can you see now?
Yeah. Can you unmute your microphone, Sergio?
We cannot see anything of your screen. You have to share again.
Oh, sorry for that. I press button like let me do it again.
And now it's, yeah, now we see.
Okay, good. Sorry for that. Like Yes. So that now you can imagine, if receptor uptake that plasmid then become transconjugates. But the question is, how could you distinguish transconjugates from the recipient or even donor? So what we did, we using transconjugate selective place. So if we add 4 antibiotics on that place, so you can imagine donor will not grow because donor is very sensitive to the CHR. So recipient will not grow further, because recipient is not resistant to other 3 antibiotics. But only transconjugates can grow on that place. So that's why we can count the number of transconjugates. Then we can use this equation to calculate a conjugation transfer frequency or ratio. So we normalize a number of transconjugates to the number of the recipient. We can go to this frequency. So now our research aims, if we add some pharmaceuticals, they are not antibiotics. So how about that process? They could be decreased or they could be increased. This is our research question. So we did the first proof of concept study. We choose carbamazepine as one of the non-antibiotic pharmaceuticals. So carbamazepine definitely is not antibiotic. It's designed for the anti-epileptic drug. So this compound is very, very robust and very, very persistent. They can't be easily biodegraded in environment. That's why we can detect carbamazepine in many different environment settings, including even groundwater, drinking water, surface water. Somehow, the concentration even up to the milligram per liter, 1 ppm above. So that's why we choose carbamazepine as a model to test our hypothesis. So what we found is, you can say, based on the transconjugate place, for control, we have very few cloning growing on it. That means spontaneous frequency is extremely low, just around 1 per 1 million cells. But if we add carbamazepine, you can say from 0.05 environmental relevant concentration to the 50 ppm, the cloning number increase. We have more and more cloning number. That is the transconjugates. So then we calculate the frequency. We found, so no matter from E. coli to E. coli or from E. coli to Pseudomonas putida, under exposure of carbamazepine, that transform conjugation transfer frequency has been significantly enhanced. So we calculate folder change. So under some condition, we also say the dose age dependent trend. In the very high concentration, we have more than 10 folder time change. So this folder change somehow even equivalent the folder change caused by antibiotics. So that means carbamazepine is not antibiotic, but they can play antibiotic-like roles. So, but you will be challenging me. Look, for this cloning growing on that place, are they real transconjugates or not? So what evidence you have? So what we did a few experiments. The first experiment, we measured the MAC based on the friendly or the randomly selected cloning. So we measured the MAC. For donor, you can say they are very resistant to these 3 antibiotics. MAC is quite high, but a little bit sensitive for CHR. But for the recipient, they have opposite trend. They are very sensitive for this 3 antibiotics, but it's a little bit resistant to the CHR. But for all the transconjugates here, so they become not only resistant CHR only, but also become multiple drug resistant. They have other resistant ability. So this is one evidence. You can say, look, after that, transconjugate harbor some gene to resist this multiple drugs. So then we also extract the plasmid from this cloning. So we run the gel. You can say for donor cell, for sure they have a plasmid, but the recipient doesn't. For all the transconjugates at the same location, same band size, they all have they harbor the RP4 plasmid. This another evidence show yes, they could be the transconjugates harboring RP4 plasmid. So after that, we want to check all the gene has been transferred from donor to recipient or not. We run the nanopore sequencing. So based on nanopore sequencing, we can say all the RP4 related gene and plasmid-based gene have been transferred from donor to the transconjugates already. So collectively, we can say carbamazepine is not antibiotics, but it did enhance conjugation frequency. So now the question becomes why? Why such no antibiotic pharmaceutical they can promote like gene transfer? So then we test the RS because I mentioned antibiotics do have RS like enhancement. So is that possible that they have the same trend or similar trend or not? So the answer is yes. So under exposure carbamazepine, you can say no matter donor cell or recipient cells, their RS per generation has been dramatically enhanced from 0.05 ppm to the very high concentration. So then we also, but this is just a positive correlation, it's not direct evidence. So then we run the scavenger, RS scavenger, we add cyrourine. So that is a chemical they can clean off or get rid of the RS radical. So then after that, RS will be gone, but correspondingly, the conjugation frequency will also drop to the control level. So that's why we can draw conclusion. RS indeed playing a very, very important role to enhance conjugation. So what else? So we also run the wide whole genome RNA sequencing. We found some RS related gene have been upregulated. This is quite consistent with our RS measurement data. So for example, SOD is one called enzyme to regulate like oxidative stress. That gene has been upregulated under carbamazepine exposure. So we also test cell membrane permeability because when we like a bacteria has a conjugation, they need to overcome that barrier. So the gene can be cell membrane will be buried for that gene transfer. So based on the TM, we can say, okay, somehow cell membrane permeability will be damaged. Like then we use FACS flow cytometry to quantify the cell membrane permeability. So the results also give our very positive trend. So under exposure carbamazepine, the cell membrane permeability has been somehow enhanced. So based on the RNA and the protein data, so we also got a very, very like similar trend. Some membrane protein have been upregulated significantly under exposure. So sum up, so carbamazepine is not antibiotics, but it could be trigger the SOS response. It could cause or trigger RS overgeneration. After that, cell membrane will be damaged, and some like plasmid-related gene or replication gene will be upregulated. So then carbamazepine can promote gene transfer from donor to recipient. So this is a first study, a concept study, but you will be further asked, okay, this phenomenon is universal or just very like drug specific or unique. So how about other drugs? We did a follow-up study. So now we choose 6 different no-antibiotic pharmaceuticals, like including anti-inflammatory drugs, ibuprofen, and we also have one drug, like one lipid-lowering drug. We have one beta blocker. We also have one contrast media. So these 6 drugs definitely are not antibiotics. It's very, very commonly used like human pharmaceuticals. So you can say based on this bar chart figure, so except this like contrast media, this purple bar, that drug can't promote like gene transfer. But for the other 5, they can promote gene transfer. For even for the ibuprofen or leptin, this like anti-inflammatory drug, they show that dose-dependent trend under high concentration, like the further change, like increase further. So we try to link why we try to address why this pharmaceutical can enhance like gene transfer. So we found for these pharmaceuticals, so they also share some common function group with antibiotics. For example, they have a carbazole function group. So that's why I guess or we assume this pharmaceutical they could cause like oxidative stress because they have such function group. They can trigger some like RS generation. So this is we further confirm this is not like unique phenomenon. It could be the universal phenomenon for many different human drugs. Okay, so the next question we want further, okay, so how about the transformation? Because we know some eDNA will be released. That eDNA could be uptaken by the other bacteria. If we dose in pharmaceutical, can that process be enhanced or not? So we built up another model with the transformation. So what we did, we choose like a free DNA is a PWH plasmid, like they harbor the two antibody, one is BRA, one is TTA. So we choose Acetobacter as a recipient. That is a competing cell. So we mix them together. So spontaneously, that gene can be uptaken by that bacteria. Then they become transformates. So now we want to test if we add no antibiotic pharmaceutical. So that process will be promoted or could be suppressed. So that's why we add like a few no antibiotic pharmaceuticals. So then, based on the selective plates, we also can distinguish like between the recipient and transformates. So we're using this equation can calculate the frequency. Very similar with our conjugation, but we're using plasmid or free DNA as a kind of donor. Okay, the results very, very similar, very, very consistent with our conjugation model. So only this compound contrast media, so it couldn't increase transformation frequency. But for other five compounds from 0.05 milligram per liter, for example, for this one, gray bar, they can also enhance transformation frequency. So the answer we demonstrate these drugs can promote gene transfer. So I would thank my one of my PhD student. She just finished her PhD student. For her topic focus on HJT, we also develop a mutation model. But today, due to the time limitation, I can't go through all the results. If you're interested, you can check through these few publications. So we want to address another question. We want to zoom in to check, okay, can we see that gene transfer or not? Can you visualize that gene transfer process or not? So because seeing is believing, as I mentioned before. So another student developed like using microfluidic system. We using like a GFP labeled donor. We also have some recipient. If that gene RP4 or other plasmid transfer from donor to recipient will show like gray fluorescence. So then, based on the confocal microscope, we can track that real-time process. So here I just show like very two short video, give you some idea. Like look, this is a control group. You can say we have a donor is red, and the black cell is recipient. After like a few minutes or 10 minutes, we do have a transconjugates generated, but the number is very, very limited. So we just have, after 2 hours, we just have a few cells become transconjugates. So this is one short video. But if we dose some pharmaceutical or some other chemicals, you can say like this process can be dramatically promoted. We have more and more like a gray cell, like they tend to gray cell fluorescence. They have fluorescent on it. So like this is some we prove or demonstrate. Yes, this is some pharmaceutical can promote from the single cell level. So after that, okay, this is just an experiment or lab-based experiment. So we will keep asking, okay, how about the gene transfer and the community level? So for example, for our wastewater treatment plant, we have very complicated like a population or community. So is that possible? These drugs can promote like a gene transfer in the community level. So we build up another model. We use active sludge as recipient. We mix one donor E. coli with RP with the plasmid labeled by GFP. We mix them together. We do the cell sorting. We try to sort out like transconjugates and recipient. So then after that we can extract the DNA from the transconjugate pool, even from the donor pool and recipient pool. So we can run the 16S RNA gene sequencing, or we can run the metagenomic sequencing to check who will become trace conjugates in active sludge matrix. So we also test these drugs. We tested 6 different drugs. For example, carbamazepine. Like we consistently say this drug or other drugs, they can promote gene transfer even in the activity sludge matrix. So we run the sequencing. We found even some human-related pathogens, such as Legionella, I try to hide here, they will become the trace conjugates. So we know Legionella can cause very, very serious, like the infection. So if we have some drugs, that process could be enhanced in the active sludge. So next question, like, is still ongoing project. So this is not related to water ecosystem, but we are wondering, because human gut, our wastewater treatment plant is look like human gut, right? So we digest contaminants from our city, whole city. This could be the gut of the whole city. But how about human gut? Because if the people regularly take antibiotic or pharmaceutical, so how about that gut? Is that possible? The gene transfer also be enhanced in the human gut. So another student, they are running, he's running like animal experiment. We want to demonstrate or prove so that gene transfer can be enhanced or is promoted or not in the animal gut. So this is an ongoing project. So in the end, I would highlight we do have some invisible factors such as no antibiotic pharmaceuticals. So they could induce mutation, but also they can promote gene transfer through HJT. So we quite enjoy to visualize like a gene transfer among bacteria. Like I think in the future we have more and more to be exploited. Even we can consider cell-to-cell arrangement. We can consider bacterial evolution, like using some like AR technology. So last but not least, so I would thank all the team members' hard work and their support. I also would acknowledge all the AQAP family members for their support. With this, thank you for your listening. I look forward to any questions and comments.
Once again, congratulations, Janwa. Unfortunately, there's no time for questions, so you'll need to send those questions by email. Janwa, I'm sure he will give you a...
Thank you.
So, congratulations again, and thank you for your presentation. This brings us to the second award, the second awardee, and this one is actually named the Arden Locket Prize. And just to remind us who these two gentlemen, sorry, this is how it was back then, who they were, Arden and Locket were working at the Manchester Davyhulme wastewater treatment plant, and they were the first ones to recognize that this thing in suspension had some biological activity, and they actually conceived the fact that by bringing this back into the basin where the wastewater entered, reactions went faster. So they are credited with having invented the activated sludge process. So, and as I already pointed out, activated sludge is also the origin of microbial ecology. That's where a lot of the microbial ecology in our field was initiated. So again, we have been awarding this prize for a number of years. The first recipient was two recipients were David Jenkins and Dick Eichelbaum, who have both left us, and there's unfortunately quite a few images here of people who have left us. Linda Blackhall got the award, Jiri Wanner got the award, Walter Tandoy got the award, Willy Verstraete got the award, and Lut Raskin got the award in 2019. So I wonder who got the award this year. It is my sincere pleasure to announce that Professor Elker Nielsen is the 2021 recipient of the Arden Locket Prize. Fortunately, there is present in Delft, and we'll walk up to the stage right now and shake my hand and then give his presentation. Give me that camera.
Yes.
Which one is it? I don't look at this.
This one? Great. So thank you so much, Bart. Thank you so much from the committee, the awardee, the award committee. It's really a great honor to be part of this great number of people we just saw. So I've been working in this field, as you know, for a long time, and I'll also come back to that. And they have really been my heroes, many of the people you just mentioned. I also worked together with many of them, and it has really been a pleasure. So to get this award, I'm really honored and very, very proud. So I promised to give a little talk, and well, we talked about should we go a little bit history or should we go about the future or whatever we should do. So I thought, well, why don't we do both? So let's take a little bit of history and also some highlights as some of the work I have done and people I've met and so on, and look a little bit forward as well. I have also chosen to do it here on site. So I'd like to thank you guys from Delft a lot to make it as a hybrid, because it's a lot more fun to speak to people and see them in the eyes than just look into a screen. So it's really nice to be here. So thanks a lot for a great conference. Let's see. I think this is the first slide. We jump back to Arden and Locket more than 100 years ago. And I think there's one key word to remember here. It is slots. It's not just slots, it is activated slots. So that was the time where we got this strange word about activated slots. So it's also alive. That's important. And that is what people realized the following many, many years, because this technology was then part of the wastewater treatments around the world. It became more and more popular. But they also met the problems that can arise running huge bioreactors. So some of the problems they especially had were to make sure you got nitrification at some point, but also filamentous bulking. That was a huge problem. And a lot of people worked with that, but there were no tools and they did not know a lot about it. And in 1988, I came also into the business. And that was at the time we formed this activated slots population dynamics group. That was in 88 in Brighton. That was my first conference ever. I had just graduated a couple of years before as a master student from University of Odense in Denmark. And as a PhD student, I was part of this group. And you can see there are many other good names. Yervana, Peter Grau. Yervana was later also head of the group for a long time, and he's also on the list, you just saw. So why did I end there? That was because I actually worked with filamentous bacteria at that time. I made my master project in that. And you can see that was at that time where you could publish a paper just as one student. So I don't see that very often anymore. I looked into this science and it was really fantastic. So I sort of fall in love with those filamentous bacteria. And I was the person in the Nordic countries and Northern Europe who knew something about filaments. So I came into this this group. Then this newsletter, it was sent out half a year later. Well, bulking was an important thing, as you can see. The chairman, he wrote, as you know, activated slots bulking has been a bestseller for quite some years, and it attracts both the universities and the private sectors and utilities to do research. And that's really attractive because it could solve everyday problems. And that's basically what I have done since, trying to help doing that. And then at that time also the nutrient removal was coming in some countries. And we had the boiling point, but not only, it was also about biodegradability, and I think we are still struggling with that. It was also about population dynamics. As he wrote, no doubt one of the highlights of of science at the end of the century. I think we can move it forward. It's still really an important thing. Mathematical modeling was also an important part. And then something many of you will probably not know why I'm reading this. Finally, something rather delicate. Are you happy with the random numbers given to filaments like 0041, 0092, and so on? Quite frankly, we are not. We have many good manuals, but there is no logic in the notation. So they want to do something, and no one else in the world can do it, but we can. So what are this guy talking about? Most of you would probably not know, but one of my heroes, that's Dick Eichelboom. He's also here from Delft. He made a fantastic investigation in the 70s, and he made a microscopy of more than 1,000 samples and tried to figure out what sort of filamentous bacteria were actually present in this. So he gave them group numbers and numbers. And that was the notation that has been used actually until very recently. So you can see at the right hand, the manual is still used. So the point was that just by looking into a microscope, because that was the only tool we had at the time, we could see how thick it is, we could see the color of the stainings and so on. We can get a name, a number in this case. So that was basically two tools. We could either make respirometry, measuring oxygen consumption rate, or we could take a microscope and do something. That was it. We knew we did not catch everything. So a few years later, I got a really exciting experience, and that was at the conference in Paris in '93. Because at that time, Mickey Wagner, he gave his first public talk about using fish. And he was working as a postdoc in Munich at that time. And that was really an eye-opener for all of us, because we had never thought about what's actually there. We knew it was something, but at this time we learned we could put colors on it and we can see the different species. And something we thought was the true, for instance, that Nitrobacter was the nitrifier, was shown is never present, it's nitrospire, and many other things. So, but this was only visualization. I really wanted to learn something about what do they do, those bacteria. And then we developed a method called microaudiography. And I know many of you younger guys do not know it. It's actually more or less still the only way where you in situ can see what bacteria are doing. You add some substrate, you label it, you can see exactly what sort of bacteria are taking it up. So it's pretty tedious to do, and I think there are maybe few labs in the world who can do it today, but it's still one of the few methods, and you should really consider if you want to see what do the bacteria actually do. At that time, we could then refer to the filaments, that this filament has eaten this or that under those conditions. And then a few years later, we teamed up with Michael Wagner again and combined it with fish. And that was the gold standard for many years to look into the microbial dark matter. So this tells us that methods is really what is driving, it has been driven the development in this field. And just to not spend all afternoon in going year by year, then those are the some of the important things that happened in the first years. This clone library by Phil Bond from UQ was important to get this full-length sequences. And later on, we got DDD, TFLP, functional genes around 2000. Holger Dimes in Munich, he made for the first time confocal. We could see 3D the structures. It's 30 years ago today now. Uresco made full cycle RNA, could could make fish probes and so on. Functional genes, probe-based, that's the place you may know if you want to make fish. That's where you, it's not updated anymore. It used to be with the newest fish probes. Isotope array, where we could combine isotopes and array stuff. Wilmus made metaproteomics in SBI reactors for Actinobacter. Isotope probing, SIP, and something really cool, the first genome, first MAC made by Garcia Martin, that was from Phil Juckenholz and Trina McMahon. That was really something that changed a lot when we got the first genome. And that was also at that time where we said, it's not only activated slots dynamic we are interested in. We want to go wider. We want to become MIVI. So we had a conference in Aalborg at that time, and we had up to that a lot of discussion about should we include digesters, drinking water, and many other things. And we decided that's the way to do it. At that time, I was the head of the specialist group, so we decided to go for MIVI, and that's as it has been ever since, and I think has been really successful to combine many things because it's very, very method driven. So so we can use it in many areas. So just to continue a little bit with the methods, we got metatranscriptomics, we got recovery of genomes, not just a single one, but many. We got the recovery of the high quality MACs. We did it together with the Vienna group, where we discovered CommonMox, combined short and long read. We made this high throughput retrieval of full-length sequences, Morten Duhon talked about yesterday. So we get millions of full-length sequences. And then we had to develop the autotags, because otherwise we couldn't make any sense of all the data. And also a new way of going into the in situ, that is to look into, use Raman microscopy to combine with fish and get information. Not the same type as this MAR, but it gives some similar possibilities. So this was a timeline for the methods related to the activated slots and population dynamics and MIVI in general. So I like to say a few words also about where are we moving. And I think some of our work here the last couple of years has been really important in getting the overview about the microbe. What sort of microbes are present out there? Based on the methods again, and this autotags, and then the global study. To all of you who helped to get those, I don't know, 700-800 samples from the world, it was really great. And I will say that now we have a name on more or less all microbes that are important in our wastewater treatment plants worldwide. And that means we have an ID, and when we have the ID, we can always link something to that. That hasn't really been possible before. And then we can compare our studies, link it to function, and whatever. So 1,500 species, that's the range, and we know many of them already. So we have gathered all this information on the Midas Field Guide, where you can go and read about what is known. So there is presently around 30,000 species mentioned in that, and around 500 are quite well described. And whenever you guys in here or somewhere online, when you publish something where you learn something about any of those species, let us know, and we'll put it in as a reference. Global abundance, as you can see at the right, there you can click at any of those countries and you can see more about what sort of bacteria are present. Here, just to go back to my pet, the filaments. Today, I would say that we do know all filamentous bacteria of any importance across the world. well, I found one a few weeks ago. I got a sample that was new, but that's probably also the only one. So if you see something that doesn't get a name or something, let me know and we will make a sequencing. But we know them all, and they have a name, and even type O21N from Eichelboom. It got the real name, Thiotrix eichelboomii. It's actually 20 years ago it got this name. But I mean, to honor Eichelboom, he did fantastic work. If we jump to the Netherlands, we can see the key contributors. That has been David and Mark, and they made sure we got some samples. And we can see the most abundant filament in the carbon removal plants is Thiotrix. So, but of course, you could look for PAOs, nitrophiles, any other things, and see what sort of bacteria are present in your country. One thing is name, another thing is function. The name is, in principle, just a tool, so we can link everything to that. The function is really what we want, either the physiology or related to performance of the plant. So I was so lucky to have a fantastic postdoc, Caitlin, she's still working with me. She's on maternal leave at the moment, but she did this great job of retrieving more than 1,000 high quality mags. And that has really changed a lot. First of all, I like to stress, as we also discussed yesterday, it's important to have high quality mags because it contains the 6NS gene. And then we can combine that with any other study, empirical study, and so on. We can also make fish probes for that one. If we don't have the 6NS genes, there's no way we can do that. So it's really essential. And we can do it now by combining short and long read sequencing. So just to get started, I think it's still more or less the only one that has been made, but I know many people are working in this. She got all those mags and they represent a lot of phyla and almost 600 different species. And if you think about, we are talking about 1,000, maybe 2,000, this is a significant fraction. And they also make up quite a significant fraction of the biomass in our plants, 30%. And if we rank them here, we can see we do have all the nitrophiles, many denitrophiles. We have all the PAOs, GAUs, filaments. So we do have most of those we know are important. So that means we can really use those for mining, get metabolic models and so on. We can also get an overview about who of those are important for degradation in the plant. Municipal plants get a lot of colloid particles. It has to be degraded. So we have tried to look into what sort of enzymes are degraded. So Ken Westman, he has spent a lot of time now looking into the secreted proteome, and we can see where are they located, all those enzymes, but we can also see which one are degrading DNA, RNA, proteins, polysaccharides, and so on. And we do see a really distinct clustering of different groups or species that do different sort of works. A lot more about this later on. I can just say that we also, the green dots growing, as we talked about yesterday, it's not all of those bacteria that are growing in the plant. Some of that's coming with influence and dying off. They are not of special interest, but all those green here are actually growing in the system and doing something. So that's cool, I think. But otherwise, I wanted to go back. It is really a, let's say, a treasure box, because we can look into anything in all those mags. The nitrophiles, we can look for common moths, we can look for new PAOs and so on. For instance, we looked into Dechloromonas because we know, or we knew some years ago, it could be a PAO. And then all the genes were present, and we could make fish probes. We could see how they look like. And then we have made a lot of studies and so on. And we can see there are two abundant species, Dechloromonas phosphoripropha and phosphorvorans. One has full DNA identification, one has not. So within a genus, we have differences in the DNA identification properties and many other properties. So that means it is important to go to a species level if we really want to know what's going on. Here it's still a metabolic model, a potential. So by combining fish and this Raman microspectroscopy, it's actually possible to verify that it has this actual physiology. So by that you can measure poly-P, glycogen, PSA in one cell, and you can also quantify. So that means if you make uptake-release experiments, we can also see where they actually release and take up, and how much. So we can actually make quantitative metabolic models on this. In this case, we could confirm that they all are important and abundant in plants in Denmark and across the world. So that sort of work are we doing now on many different groups, and I know other people are doing the same. So I think we will pretty soon get a really nice overview about most of the bacteria. So the approach, just to sum up, the approach we're using is that we are, first of all, we have those high quality mags we can use for metabolic reconstruction. But then we have the 6NS amplicon time series. We have the reference database and the global. So we can also see where they are abundant in some countries, some plants, and so on. And then we can produce those models and confirm it by Raman. And we can then use that information to follow in the plants. And of course, get new samples if something is missing. So this is the concept we're working with. And from an academic point of view, this is really cool. We learn a lot. But then someone would ask, I'm sure David will ask, so how do we transfer this to practice? And that's what we're doing in this Midas wastewater treatment plant collaboration. For 15 years now, we have collaborated to many Danish plants. In the beginning, we just got samples once in a while, we made fish and so on. But the past 5 years, all of the plants have collected samples every week. And we have now sequenced all of it, so we have time series for all those plants for 5 years. Also digesters actually, by the way, not only actual slots. And then all the plants have online access, so they can simply on the computer or the app see how do the different bacteria act or change over time. So right now we are building up experience. They are learning. We call it know your own plant, and so they learn if you do this, then this happens, and if you have a problem, was it due to some bacteria, and so on. The only problem is that we get the data some months later. So we only sequence once in a while. And the solution here is, of course, to do it right away on site. So ideally, sample, generate data, identify microbes, get the information about whether they're important or not, and then act if something is outside your level. And that's what we have done, and as what we're doing. So Martin and Mads here, they have already done that several years at our local treatment plant. You can see when you sequence, it just takes 30 minutes to get a stable signal. So it is possible to get data the same day or from day to day. And you get immediately the identity and so on. So we have done it now for one plant where we have tried to control sludge age for a year. And we have done it from a demon reactor where we have measured every second or third day for a full year to control such an anamorph process. So it's working, and our plants will now test it full scale in larger scale next year. So with that, I think we are closing the loop. When I started in '88 with this specialist group, the activated slots population dynamics, the goal was to know what sort of bacteria are present, what do they do, how can we manage. It took a while to get all the names, but they are in the database now. We know a lot about the function, and we are also ready to go out and use it. We can already do a lot of stuff, but I really think that the next few years is moving forward. And so I really think it's a very light future for the activate slot system, wastewater treatment system, but this can be transferred also to any other system. So a light future for this and for the field. I think there's so many things are moving forward. So many people are working in the field. So I'm really happy about that. So nice to see, and I like to thank a lot all the people in the back home, Center for Microbial Communities. They are, of course, the people who are actually doing the stuff. And there have been a lot of people around for the past many years. And I also like to thank all the foundations and also all those who sent samples to us. And finally, I think I like to thank another time for the award. It was really a great honor, and thank you for you guys sitting here, and nice to see someone alive. And thank you to all you guys online. I hope you enjoyed the talk. Thank you.
You still have something on the slides?
It's okay. I can just speak. Well, congratulations again, Per. I cannot think of a more worthy recipient of this award. 1988, it's actually only been 30-something years. It's been amazing, and you have been a major contributor to all of that in the last 30 years. Very, very nice. Very well deserved. So this brings us to the announcement of where MUI 10 or MUI 2023 will happen. And just to give you a minute of background, so this is actually the first time MUI 9 or MUI 2021 when we organize the conference biannually. Before it was every 3 years. This is the first time there's only been a 2-year gap between Hiroshima, MUI 8 and MUI 9. And when we called for the conferences in 2019, we called for both MUI 9 and MUI 10 at the same time. And we will do so again the next time, because it always takes a lot of preparation to submit your proposal. And rather than just having one winner, we decided to choose 2 winners for the 2 sequential conferences. Before I announce where 2023 is going to happen, I want to actually use this opportunity, because this is the last time I'm at the podium, to thank the 2 organizers, both Mark, but especially David, who seems to have been living this conference for a year plus, once again, for their amazing work to pull this off. So give them a big hand of applause. And so this brings us to announcing where 2023 will happen. After I say my word, somebody will launch them, please. So MUI 10 or MUI 2023 will be hosted by the University of Queensland in Australia, together, and both Janwa Guo, who you just heard, and Adrian Ullman are the co-organizers. Congratulations, and see you all in 2023 in Australia.
Thank you so much.
It's this, right? Yes, exactly.
Yeah. Good morning, good afternoon, and good evening. It's our great honor to host the 10th Microbial Ecology and Water Engineering Specialist Conference in Brisbane in 2023. My name is Jianhua Guo. I'm Associate Professor at the University of Queensland. I will be also the chairperson for this meeting 2023. So in the following few minutes, my colleague, Dr. Adrian Oman, and Dr. Cedric Hall, plus myself. So we would like to give you a very brief introduction about our idea, program design, and aims of MIVI 2023. We also would like to introduce our beautiful city, Brisbane, to all of you. At the beginning, so we would like to make acknowledgement of country. So here at UQ, we acknowledge the traditional owners and their custodianship of the lands on which we will meet. On behalf of a local conference committee for MIVI 2023, I pay our respects to their ancestors and their descendants who continue cultural and spiritual connection to country. We recognize their valuable contributions to Australia and global society. Actually, as one of the IWA specialist groups, so our MIVI specialist group has long history until now already. So initially, in 1988, the specialist group on actively large population dynamics, SPD, was formally established. So now you can imagine in 2023, we will celebrate the 35th anniversary of establishment of the MIVI specialist group. So in the last few decades, actually, we have organized 9 MIVI conferences already. So we have another milestone to celebrate. So in 2023, we will have our 10th MIVI conference. So that's why I hope all of you can join with us to celebrate such 2 major milestones in Brisbane. So this conference, we are aiming to boost up our communication among universities, research institutes, and water industry. So we hope we could facilitate and promote synergies between fundamental and applied research. So actually, our university has put a lot of efforts in to enhance collaboration between industry and academic. So one example I want to share with you is in Brisbane, Queensland Urban Trinity and our university built up innovation center at Locket Port with wastewater treatment plant, which is one of the largest wastewater treatment plants. So over there, we built up quite a few pilot scale bioreactors. We aim to demonstrate new technologies and new processes. We also try to use some microbial ecology toolbox to guide us to further optimize reactor performance. So as shown in this figure, so we build up very large scale silver networks. So we are also exploring some microbial ecology related topics based on such very decent facilities.
Okay, hi everyone. So when planning out the program for the MIVI 2023 conference, we wanted to have a mix of different themes, some core themes within the space of microbial ecology and water engineering, as well as several emerging themes as well. So as Jianhua mentioned, one key topic we want to focus on as well is linking microbial ecology to process performance in water engineering systems. And so that link between research and application, and also several other emerging areas such as the ecology and diversity of viruses, bacteriophage in water engineering systems, to name a few, as well as a number of key topics, both application-wise and methodology-wise regarding microbial ecology and water engineering. Okay, so next slide. So we have put together an organizing committee for MIVI 2023 that we think really combines expertise within academia and industry. And so we've been looking, we have colleagues within a number of the utilities, for example, like Jianhua mentioned, as well as a number of universities, certainly the University of Queensland, also some other universities within Australia, and research groups and companies such as Aquatech as well. Okay, and together I think we provide really the core expertise needed to have a good coverage of all the different relevant topics with that's covered within the MIWI specialist group. Okay.
Thanks, Adrian. And I'm Sandra Hall. I'm the General Manager of ACWEB, but I'm also a Director of the Australian Water Association and a Fellow of the IWA. The Australian water industry is a thriving, diverse and collaborative sector. It's a sector that has an annual revenue of about $24 billion and employs 28,000 people across the country. This week is actually National Water Week, and we're focusing on caring for our water and our country. In the Southeast Queensland, the partnerships between utilities, innovation providers, universities, consultants, and our communities is driving the research forward, ensuring that we have a sustainable water future. I am a Brisbane born and bred girl, and I'd love to have the opportunity to bring you here to our country where we have access to some of the most pristine environments in the world. Let me introduce you to Brisbane. We'll run a video.
Take everything you need, take everything you see, though. Take everything but me. Sit your presence next to mine, but you know I will not resign, 'cause at your top is hard. But the river still flows from your body, your toes, and it's filled with all your thoughts that my body ever knows. I thought that you'd be the one to be there for me, but obviously my thoughts and views of you were wrong, 'cause that's my job. You hit on, you gave it back, and now you're gone. Play my heart just like a frustrated in those pants that you are wearing. Tie me up on what's become undone. Sitting in the passenger seat, feeding me what you know, Cardi. I spit the line and then I'll come. But the river still flows from the heart of your toes, and it's filled with all your thoughts that nobody ever knows. I thought that you'd be the one to be there for me, but obviously my thoughts and views of you were wrong, 'cause that's my job. You hit on, you gave it back, and now you're gone. Oh, yes, now you're gone. Oh, now you're gone. Oh, now you're gone. I thought that you'd be the one to be there for me, but obviously obviously my thoughts and views. But you are wrong, 'cause that's my job. You bet I thought that you'd be the one to be there for me. But obviously my thoughts and views. But you are wrong, 'cause that's my job. You bet I gave it back, now you're gone.
Thank you for listening. So me and Wei look forward to see you, all of you in Brisbane in 2023. Thank you one more time.
Congratulations, Brisbane, for this, and wish you all the best for the organization. And hopefully we can meet in person in 2023. But are you leaving already? Yeah. Thank you so much for having been with us on site as well. And all the best for your travel back to Denmark. Thanks. And we still have one small part in the recreational part. It's the poster awards. And so it means we do the poster awards and then maybe a short break of 5 minutes. And then we start the session 8 with the keynote of Ade from Ethiopia. So let's go to the poster awards. So for the poster awards, we had Nicole had set a platform where all the participants could join in and vote for their preferred posters across the different sessions. So it was a peer evaluation of the posters, and there were 2 posters that were recognized. And I will give the names and will display the certificate. You will receive also a present here that I have in hands and that we will send you per post to your home address. Let me advertise the posters that are selected as certificate. And the first poster comes from Kaust in, how can I make full screen? Display, full screen mode, yeah. So the first, there are 2 poster awards and equal quality, and one of these poster awards is delivered to Changshi Wang from KAUST, King Abdullah University of Science and Technology in Saudi Arabia, for the poster on metagenomic investigation of antibiotic-resistant genes and virulent factor genes in extracellular DNA across wastewater treatment processes. We saw this morning in a talk from David Calderon that eDNA is a part that has to be consistently addressed in the future. Congratulations, Shang-Chi. I don't know if you are online, but warmest applause to Shang-Chi Wang. Are you online, Shang-Chi? No, but we will give you a present that I will show after the second. And the second poster is this delivered to Francesca Pretilieri from Aalborg University on Candidatus Acuminibacter. Refined phylogeny reveals the existence of novel species, their potential function, and their global distribution. There is a saga of PAOs, as per explained, and Acuminibacter is always rediscovered with all these techniques. So congratulations, Francesca, for your work. So what you will receive, Francesca and Shang-Chi, it's a present from Royal Delft. So in Delft, we have the Delft blue porcelain, and this will be a plate that you will receive. Maybe I can show it to you already, if I don't break it. I received the poster award in Shanghai, and my son played football at home, and the award got broken, and I had to glue it, and I still have it. But this is the award from Delft Blue. This is a map of Delft, actually, on a Delft Blue porcelain of Delft. You see here the channel. This is the port of Delft, and we are around here on the map. So this is what we will send you together with the poster certificate award. So we will need your addresses to send you that. Congratulations, Francesca and Changxi again. Yes, so I stop here the the recreational part. I will acknowledge Nicole Romy and all the PhD candidates at the end of the day, as soon as we will be completed with session 8 and 9, so the 2 last sessions of the conference. I will let Mark and Francesc chair the next session with the presentation of IDE and other colleagues on water and health engineering. See you later. Pardon? You have to change now to the platform. We can make just 5 minutes to make the transfer of the computer, etc. But so in 5 minutes we start. So just join the next session directly now in your poster, in your platform. Thanks.